Hi, audience. You're relaxed? I think the Mozart effect has started because one says that after listening to good music, you will pay more attention to my talk. Aha. So, why am I here? My topic today is about brain cancer and why research is so important in Malaysia to find a cure for it. Why is brain cancer important? Because it affects 1,500 to 2,000 people per year in Malaysia. The calculation is about 5 brain cancers a day. Just imagine as we are talking, there is one doctor telling his patient, "Dear sir, dear ma'am, you have a brain cancer." And whether this brain cancer is curable or incurable would be another question. We both know that there are 2 types of brain cancer. We have the benign type, which is curable, and we have the malignant type, which is difficult to treat. So, as many patients that came to my doors asking for help, I had problems trying to tell them that the treatment for brain cancer hasn't changed very much since I did my residency 20 years ago in Belgium. It's still about the same. So, and most of these patients succumb after a period of 24 months. So, after I cried with my first patient's family who died, I kept on crying till I had no more tears left. So I decided to do something. I decided that I would need to find something to kill this brain cancer cell. So what did I focus on? I focused on the most malignant brain cancer known to the human being, the glioblastoma multiforme, or better known by the World Health Organization as the grade 4 glioma. So I was reading and reading, what else could we do? Have we run out of bullets? Have we no more things left in this world to treat this type of cancers? And I found out that we might be able to use a virus to treat cancers. Many people are using virus. You're using herpes simplex virus, you're using HIV virus, and some people are using very infective virus, but you must remember that these viruses are also dangerous to us. So I found out that in 1929, millions of chickens died in a small little town called Newcastle in the United Kingdom. And someone had an idea. In 1950, Laszlo Kasari, a Hungarian physician, took this virus, live virus, and gave it to his patients because one Hungarian chicken farmer who had a terminal stomach cancer was totally cured of his cancer by just accidentally being exposed to the virus. So the story goes on. I was thinking, what could I do? And I found out within Malaysia's shores that University Putra Malaysia had a Malaysian-made Newcastle disease virus, V4UPM. And therefore, since 2009, our lab started doing experiments with V4UPM to see whether it would be able to kill these cancer cells. The reality of doing experiments in Malaysia is that when we discover technology, it should be accessible, it should be cost-effective, it should be affordable. Just imagine finding a cure for cancer, but it costs the patient 4 million ringgit for the patient to be cured of his cancer. That would mean a disaster for our Malaysian population. And number 2 is the virus that we have to use for the patient would be a simple virus that could be used in any hospital, whether it is in East Malaysia or West Malaysia, without infecting the researcher or killing the patient. So, let's go back to the story of this Newcastle disease virus. What is this virus? This virus comes from the order Mononegavirales, the family Paramyxoviridae, and the genus Avilavirus. Unlike its cousins, the HIV virus and herpes simplex virus, like I said, which does not infect the patients who are exposed to it. The NDV virus has 3 strains. It has the most virulent strain called the velogenic strain, and it has the least lantogenic strain, which does not kill the chickens, but just make them sick. So this would be a perfect virus to be used in Malaysia. We do not need to build million-dollar labs, isolation, blah, blah, blah. And we could be able to use this virus in most hospitals, maybe in the near future. How does it work? So in our research, in our published research, the glioma cells are highly invasive. They kill patients pretty quickly because of this mutation of this protein called RAC1. And our experiments have shown that the NDV from Malaysia goes straight and kill these cancer cells because of expression of this protein. So voila, we have found a key, a way to kill these cancer cells. So what did we do? With collaborators from UPM, we took the V4 UPM. We had to grow this in embryonated chicken eggs so that we could then harvest these viruses to be used in experiments in our lab. So, after 4 papers and chapters in textbooks and expenditure of about 1.5 million Malaysian ringgit, we finally came to one conclusion, that a brain tumor the size of a small golf ball or small egg would need 3 million infectious particles. And one chicken egg, is able to produce 500 million infectious particles. Therefore, 6 eggs could be used to produce enough virus to destroy the golf ball tumor in the brain of that patient. So, how is it done? It is done by doing it in biosafety cabinets class 3, in a special room, but not expensive isolation labs that we would need for more important and more dangerous virus like HIV virus or herpes simplex virus, where the virus is seeded and taken to a process from candling to purification so that we would be able to harvest enough of this pure virus. So look at this slide. Look at how the virus gobbles and kills and blabs and blows the cell. Wow! You know, look at it. When I was looking this under the live microscope, I felt so happy. And then we did special staining, and it showed that the cytoskeleton of the glioma cells were totally destroyed without destroying the normal human brain cells. That was most important. So, let's smile. Let's think. What we are doing currently, treating brain tumors, gliomas, with radiotherapy, with intravenous and oral chemotherapy, and also intraoperative chemotherapy, may be helped one day by using viruses from an egg, a Malaysian egg and a Malaysian virus. So I think one day there might be still hope. So, life hacks. Well, the animal's enemy is a human being's friend. So one day you might think that, you know, you can use someone's rubbish to make someone else gold. So that's one important thing. And Malaysian Newcastle disease virus strains has been proven to kill the human brain cancer in rodents that we have tested, and also in ex vivo cancer models. I took 30 cc of brain tumor out from a patient, transported to the lab, and injected the virus into this block of 30 cc brain tumor. We showed that it died after exposure to the NDV virus. But further experiments are needed, as well as clinical trials. I would like to thank my collaborators listed here from USM, University Putra Malaysia, and the help of Magna, who has helped us tremendously. And we, as we speak today, we are still fighting and fighting, and we will not give up. And I think what is important for us, we hope to find a cure for this malignant brain cancer within the next 5 years. Thank you very much.